from South America, Asia, and UK, seeking new therapeutic solutions to leishmaniasis and other tropical diseases. Both COMSTECH and NDT network aim to promote global scientific cooperation and build researcher capacity to find new and better therapeutic solutions to these neglected tropical diseases. The second lecture will be delivered by Professor Dr. Paul William Denny. Professor Paul is a full-time professor at University of Durham, United Kingdom, and under Department of Biosciences. The title of his talk today is Protozoan Drug Discovery, Old Drugs, New Targets, and Mining for the Novelty. With this, I would request His Excellency, Professor Dr. Iqbal Chaudhry, Coordinator General COMSTECH, and Director ICCBS to kindly introduce and welcome our guest speaker today.
Thank you, Khazima. Thank you for presenting the key objective of this lecture series. We are absolutely delighted to partner with NTD. We have been associated as an institution with NTD since last several years, and I met with Professor Paul Denny in 2017, I believe, and it was a wonderful meeting where we constituted this extremely important network focusing on understanding the molecular basis of diseases such as leishmaniasis and Chagas. Paul is truly and genuinely someone who is motivated to develop capacity in the world when it comes to science and skills, and he has been heavily engaged with many institutions of the world and many networks. We were very fortunate to host him in Karachi pre-COVID time, and he conducted an excellent workshop in which we have invited people from other countries also. And the emphasis of our discussion through our various meetings was that we need to go beyond the network countries, the 6 countries. And that problem is of a global nature, which requires global actions and global alliances. And this nexus between the network and COMSTECH is precisely a step towards that. And as I was discussing with Professor Denny, that beyond this lecture series, COMSTECH would like to continue this collaboration for the benefit of people where these diseases are so endemic and affecting the life of people in a very profound manner. Professor Paul William Denny is a professor in the Department of Bioscience in Durham University. I visited his laboratory. Excellent laboratory, very productive and extremely innovative. Before his appointment in 2004 as a professor, Paul was employed in postdoctoral associate and research fellow posts with Professor Debbie Smith. I believe this is Professor Deborah Smith of Imperial College. A very well-known figure in the field of tropical diseases and drug development, someone of which made a seminal contribution in various fields of tropical disease treatment. Paul developed their interest in the molecular and cell biology of lipid biosynthesis in kinetoplastid protozoan parasites, particularly leishmaniasis. Now, with a colleague at Durham University, Professor Paul is focused on characterization of protozoan sphingoid biosynthesis and the identification of new antimicrobial targets within both the Kinetoplastidae family and Apicomplexa. He is the founding director of Durham Center for Global Infectious Diseases, a very important initiative. He is also a principal investigator and director of Durham Global Network for Neglected Tropical Diseases and team lead for the Molecular Science for Medicine. This is EPSRC Center for Doctoral Training, Newcastle and Durham. Several examples of Paul's scientific work on the inhibitors of leishmaniasis sphingoid biosynthesis have recently been published and cited. Furthermore, he has also been extensively involved in omics science, particularly targeting to understand new anti-leishmanial drug targets, which is extremely important because these two diseases, unfortunately, are were neglected in a true sense, and we know very little about these parasites, and thus we're not able to develop successful drug. With this, I would like to invite Professor Denny for his presentation.
Thank you, Iqbal, for those very kind words. It's a pleasure to be here. So can you see my slides?
Yes.
Yeah, good. Okay, so thank you. So I will begin. So yeah, to me, this is a very important initiative, engaging with COMSTECH. As Iqbal said, we have known each other for a number of years now, and we are involved in a global initiative. But it's very important that we reach beyond that consortium out to other countries and individuals who are interested in and indeed suffering the burden of the diseases that we're trying to mitigate. So yeah, I'm in the Department of Biosciences at Durham within the Biomolecular Interactions group. And probably more important than my institutional job is being involved in the leadership of the Global Network for Neglected Tropical Diseases. So as Iqbal said, we have Latin American countries, South Asian countries, Durham and York in the UK, and of course the ICCBS in Karachi. So in our first phase, which runs now until March 2022, the primary aim, not the sole aim, the primary aim is to identify new molecular targets which are essential for the parasite that can be exploited in drug therapy. So just to talk about sphingolipids, which Iqbal mentioned in the kind introduction. So they're implicated in mammalian cells, other eukaryotic cells, and protozoa in such things as trafficking, moving proteins and lipids around, and signal transduction, and also in these lipid rafts. This is a scheme here sketched by Kai Siemens, who was instrumental in the characterization of these microdomains on the plasma membrane of eukaryotic cells. So within these, we have glycosphingolipids in humans and other mammals, sphingomyelin, and they attract and harbor GPI, glycophosphorylnositol anchored proteins, and acyl anchored proteins. So proteins with lipids on fatty acids. So briefly, I'll talk about toxoplasma. I mean, it's a model apicomplexan and is a parasite of any nucleated cell. It's a beautiful parasite, to me anyway. It has this apical complex, which you can see in this schematic here, and you can see elements here in this electron micrograph. Its definitive host is cats, is a global parasite that probably infects 50% of humans, at least. It can cause severe neurological problems, particularly in the immune suppressed and the prenatal. And it's related to the Plasmodium parasite, which causes malaria. So it's functioning as a model apicomplexan, a model for the malaria parasite and other apicomplexan parasites as well. So serine palmitoyl transferase, or SPT, is the first enzyme in sphingolipid biosynthesis in eukaryotic cells, including humans. And in humans and all other eukaryotes studied up to the point that we started looking at this, it has a dimeric core, 2 units, LCB1 and LCB2, which you can see there. And that's the regulatory step. And we'll talk more about that a little bit later. Now, some bacteria, not all bacteria, some bacteria also have an SPT, a serine palmitoyl transferase, but that is completely different. So this is an example of parallel evolution. It's evolved differently towards a similar purpose. It's soluble and it's homodimeric. And our finding was that the apicomplexa, unlike every single other eukaryotic cell lineage, has a bacterial-like serine palmitoyltransferase. So here it is modeled within a SAX, small angle X-ray scattering envelope, which gives us an idea of the size and shape. And it's a homodimer, which has a similar structure to the bacterial enzyme, and evolutionarily is related. And of course, we published that. But really, I'm here to talk about leishmaniasis. Now, as many of you have been aware, there's a continuing problem with leishmaniasis across many countries, which you can see in this map here, including large parts of Asia, Africa, and Latin America, as well as Southern Europe as well. So it's in more than 90 countries. It's endemic, over 1 million new cases annually. That's a massive underestimation as well. Well over 12 million people infected. And the estimates are anywhere between 350 million and 1 billion people living at risk of disease. Spread by sandflies. Now, pre-COVID pandemic, of course, I had the great pleasure, as you heard, of traveling around, going to places as diverse as Brazil, India, and Pakistan. And on my trip to India in 2019, I picked up a newspaper, The Hindu, and saw this picture. And this is a picture of a teenage boy in Bihar, in West Bengal, suffering from Kala Azar, black fever in Hindi, and it's the disease that just won't go away, and that is visceral leishmaniasis. And closer, and this is something that Sama organized when I had the pleasure of visiting ICCBS, is I had an opportunity with Mags Layton to go to the Jinnah Hospital in Karachi, to the dermatology ward, and I actually met with Mags and talked to the clinicians. These 4 children from one family who had travelled to northeast Pakistan to see family before travelling back to Karachi, and they all suffered from various lesions caused by cutaneous leishmaniasis. Okay, and the effect on this, of course, was scarring, but also social and educational exclusion. They weren't allowed to attend school. This is not infectious, but they were excluded from their education, and their daily wage father was unable to work. So the family suffered economically, and their children suffered educationally and socially. So this really focused my mind more than anything I think I've experienced on the disease, and particularly cutaneous disease. So as I mentioned, sandflies carry the disease, so they inject a promastigo flagellated form into the human or other mammal. They are phagocytosed by macrophages, part of our immune system. And depending on the species of parasite, the site of the bite, the immune status of the host, they transform into amastigotes within the macrophages and cause a range of diseases, visceral disease, mucocutaneous disease, that's exclusively in Latin America, and cutaneous disease, which is by far the most common, and the one I mentioned, the 4 kids in Karachi. And these amastigotes multiply, cause ultimately inflammation and tissue damage until they are taken up in another sandfly meal, and then the cycle begins again. So current treatments exceedingly limited. The top line here, this is what those 4 kids were being treated with, these antimonials, so heavy metal drugs that are about 100 years old now. They have not been developed, they're highly toxic, they can only be used by intramuscular injection or into the lesion where the parasites are, exceedingly painful and unpleasant. Developments have been particularly amphotericin B, an antifungal, in a liposomal formulation, is now being used for visceral disease in an elimination program fairly effectively. Paramomycin, an antibiotic, repurposed. Pentamidine, again, sort of generic antiprotozoal, repurposed. and miltefosine. And it's only miltefosine which has problems with resistance and some toxicity issues, but that is the only one of these 5 classes of drug that is actually available as a tablet. The rest are injected over a period of time. So we have looked at the mode of action of these. The mode of action of miltefosine remains unclear, but there does seem, this is work done with Mike Barrett and Coral Barras in Glasgow and Barcelona respectively. And it does seem that sphingolipids and related biophysically related sterols and their metabolism underlie the sensitivity of leishmania to miltefosine. So that is something we are investigating. But let's talk about the sphingolipids themselves. So the base sphingolipid is ceramide, which you can see here. It has this amide link here, which you can see, and then a fatty acyl group, and it has another fatty acyl group there. Now, commonly it'll be C34, C36 in most eukaryotes. That's 34, 36 carbons. And at this primary hydroxyl group here, an addition of, for example, phosphocholine, which you can see here, in humans, for example, produces sphingomyelin. In fungi and plants, phosphocholine is replaced by phosphoinositol, the sugar group you can see here, and that's a rather specific lipid species. Okay, there are many commonalities. We spoke about serine palmitoyltransferase in the AP complexer. That's an exception. In all other eukaryotes, this is the classic heterodimeric form you see in humans that I spoke about at the beginning. However, the divergence comes after the formation of ceramide, where we have sphingomyelin synthase producing sphingomyelin, which I mentioned earlier, by the addition of phosphocholine from the lipid phosphatidylcholine onto ceramide. Now, yeast plants, and importantly for my talk, some protozoa, including leishmania, they have inositol phosphoryl ceramide, or IPC synthase, known as all 1P in yeast, where instead of phosphocholine, phosphoinositol from the lipid phosphatidylinositol is added to ceramide to produce IPC. So this is leishmania, an image I produced many years ago. You can see the plasma membrane highlighted with a green fluorescent protein construct. This is called the flagella pocket. That's where all traffic leaves the leishmania and all incoming traffic comes in. The nucleus and the characteristic kinetoplast, the mitochondrial DNA of the kinetoplastid parasites such as leishmania. And this is labeled with ceramide. It's a red fluorescent ceramide. And this is the Golgi apparatus. So complex sphingolipids formed from ceramide and, for example, phosphoinositol, in the case of leishmania, are formed in the Golgi very close to the flagella pocket. Now, how does this happen? Well, the mode of action of the enzyme is that we take phosphocholine from phosphatidylinositol, and it's added to this activated histidine we can see in the enzyme to produce this form. This is a dynamic form. It'll phosphorylate and then dephosphorylate. And then ultimately ceramide comes in and it captures that phosphoinositol to produce IPC, which you can see here. Now, the important part here is this enzyme lies at a tipping point between the production of diacylglycerol, or DAG, and that's a growth promoting or mitogenic factor, and the diminishing of ceramide, which is a cytotoxic, pro-apoptotic factor. So it really is the balance between life and death, and that was our initial thought as to why it's an attractive drug target for leishmania. Now, the fungal IPC synthase is a validated drug target in many species. And there's a number of natural products which are have been identified as inhibitors of this. And the most developed is Aureobacidin A, which gave its name to the fungal IPC synthase. That's why it's called All-1. This has not quite made it to clinic, but has been the closest to all of these. Now, before I sort of go on, I just want to talk about high throughput drug screening for drug discovery, because I'm going to talk about all of these platforms into varying levels, and there's a number of things to consider. So the most common, and ones maybe that most of you have heard of, is in vitro high throughput screening. That's really where you take a target, typically a protein, and you have a functional sensitive assay system, and you throw compounds at it, and you get a reaction and get an inhibition. And the advantage of that, it can be very simple, very, very sensitive, and it can facilitate structure-activity relationships, or SAR, because we can have various similar compounds showing various effects on the activity we're looking at. But it has a number of disadvantages. It requires typically a pure protein, and that can be particularly challenging, particularly with complex proteins, such as those that cross the plasma membrane. It can be expensive because of the need for purification, and indeed the substrates can be very expensive. So it has disadvantages and can be very complex. Rather simpler, at least in theory, a phenotypic high throughput screening, which has gained a lot of traction, as I'll come back to. So you don't need a purified target for that. You just essentially throw the compounds at the cell type you're interested in. It could be leishmania, and it allows you to select drug-like compounds, the ones that actually do inhibit cellular growth. And we're not making any assumptions. It's a naive approach. We're looking at everything in an unbiased manner. We have no specific target. Has some problems. Can be quite insensitive because many cells, including leishmania, have pumps to remove drugs and other chemicals from them. It can be expensive to culture leishmania and many other cell types. There's no target specificity. It's just a phenotypic output. So SAR is not assisted by target knowledge. And then we have to go through quite a difficult process of target deconvolution after we have identified a compound, which is expensive and challenging. But again, I'll come back to that. But what I'm going to talk about now is yeast-based systems, yeast-based high throughput systems. So again, you don't need a purified target, but essentially you put your target into yeast, baker's yeast, Saccharomyces cerevisiae, because they're easy to manipulate and grow. And we can actually have, we can manipulate them to have a reporter platform, so we can see fluorescently or otherwise a signal on the inhibition of our target. This makes it easy to get rid of false positive hits relatively, and also they're very cheap to grow and manipulate. Now this sounds fantastic, but there are still several disadvantages. Again, they have particularly aggressive drug pumps and membrane barriers, including a thick cell wall, which can make them insensitive. Your protein often has to be functional in this foreign system. That is a limitation. Not every protein is going to be functional in Saccharomyces. And still, this target is not tested in its native state. It's in a non-native cellular environment. It's in yeast. It's not in leishmania or whatever your target organism is. But several of you, I guess, will be playing with designing assays and developing an idea of using those to screen for compounds. There's a few basic rules which we should all follow, which are often not followed, to be honest, including by me, but we should all do that. We should consider the validity of the screen, you know. Is it a valid target if it's an in vitro scheme? Are we looking at the right stage if it's a phenotypic screen? Are we looking at the pathogenic form? we need to consider the cost. In all environments, we need to consider the cost. There's not infinite money in any environment, so it needs to be relatively cheap. This is the one that often is problematic, at least in academic environments, is the linearity. So the example here, there's two life cycle stages, the insect stage and the mammalian stage. Now, obviously we focus on the mammalian stage, but the rules are the same. And we need to get the parameters right. So we have this linear relationship between the signal and in this case, the number of parasites. So if we inhibit it, we can have a signal that relates directly to parasite number. If we don't get the parameters of the assay correct, we incubate for too long, for example, and then we get this curve, and then that linearity breaks down and our assay becomes useless in certain situations. Coupled to this, we need reproducibility, of course. We don't just do things once, we do them multiple times with technical and biological repeats. And an important point is it needs to be transportable. There really is no point having an assay that we cannot reproduce in another lab on another continent. This is a real common problem, and I don't think we've got to the bottom of many instances where we cannot reproduce data from one lab to another or one continent to another. Okay, so beyond that, those basic parameters, we need a robust assay. And this is where Z prime or Z factor come in. Rather simply, the Z factor is 1, and you subtract from that 3 times the difference of the standard deviation of a positive and negative control. The positive control is a known inhibitor, a drug. Negative control is untreated, just with the vehicle which carries the drug in your positive control, typically DMSO. And that is divided by the difference between the mean of that positive and negative. Okay, so just give you an example here. So we have the equation, and then we have the data here. This is some real data produced in my lab. So we have the mean of the positive control and the mean of the negative control. So when treated, the signal is lower in the positive, of course. It's a known inhibitor. We have the standard deviation of both, and then we can start putting it into that equation. So 3 times the standard deviations, the sum of, and then the difference between the means. So we get 1 minus 1281 divided by 15440. And that gives a Z prime or Z factor of 0.92. Now, in an industrial context, anything above 0.5 is regarded as a robust assay. So in this context, this is a very robust assay. So that's something we apply at various stages in our process, but particularly in our screening process. So I'm just going to talk now about work we've done in collaboration with GSK some years ago, using yeast as a system to look at the IPC synthase from Leishmania, which I spoke about earlier. This is a transmembrane protein, which is very, very difficult to assay in vitro. So yeast, as I mentioned, OR1 is the IPC synthase. So in this line here, the OR1 has been deleted from the chromosome. It's essential, so it's supported on a plasmid with a uracil synthase selection, so it can grow in the absence of uracil. This just emphasizes really the flexibility of yeast. It's very easy to work with. So if we add another plasmid, this is Lu2, so it can grow in the absence of leucine, encoding the Leishmania IPC synthase, but it's under the control of a galactose promoter. So it needs galactose present and glucose absent to actually be expressed. And we have an episomal heterozygote, so it has both. And then a neat trick in yeast, we can add 5-fluorontic acid, and the action of Ura3 produces a toxic product, which means that the cells are purged of Ura3. And if we grow in the presence of galactose in the absence of glucose, then we get a complemented strain. And then we can show this is correct by then looking at the inhibitory effect of glucose, which indeed prevent growth of the yeast. So that was the creation of the strain. And we set it up in a fluorescent assay, which I won't talk about in any detail, but the assay allowed a fluorescent sensitive readout of growth. And then at Chescantos, funded by the Open Lab Foundation, we're working in the GSK unit there. We screened their library, which is about 2 million compounds at a single concentration against those yeast. And then we wanted to know whether they affected the Leishmania enzyme specifically, or whether it was just an antifungal effect. And we screened them at 2 concentrations against the yeast complemented with the Leishmania enzyme and also with the wild type parental enzyme. And then we got down to 4,000 compounds. And then at that point, it's a dose response screen against both these yeasts, which I'll show you now. So essentially what we're trying to do is get rid of the non-active ones, of course, but we want to get rid of the ones that are active against all one, but not the Leishmania enzyme, because these are obviously not going to be on target. And we want to get rid of the ones that are active against both. Now, why do we do this? Because chances are these are antifungals. So to get selectivity, this is our measure of selectivity, it's the activity against the Leishmania enzyme in the yeast as opposed to the parental all-1 yeast enzyme. Okay, so we're looking at target-specific compounds. Now, this is the 4,000 compounds plotted on this scattergram here. And we have the IC50, the 50% inhibition concentration against yeast on this axis and against the Leishmania enzyme on this axis. We've already gone a pre-selection, so most of them are to the right-hand side of this diagonal here and are more active against the Leishmania because this is after our second round selection. However, in this dose response, we want those that are in this corner here, the most selective. These are all log figures. So this is 1 micromolar and this is 100 nanomolar. And similarly, the ratios here, the selectivity ratios are log figures. So this is about 30 times more selective for the Leishmania than the all-1 from the yeast. So we want the most selective. So ultimately, we've got down to 500 compounds that selectively, in our assay, inhibit the Leishmania enzyme. Then we go through a process. This is an industrial process of clustering. This is just so we don't waste our time looking at highly related compounds, because libraries tend to come with clusters of compounds which are very closely related. So we look at only 2 compounds from each cluster and all the singletons that we identify, the ones that are unique, at least in this way. So then this is the first time in this process, we actually see the parasite, and we're looking at azenic Leishmania donovani amastigotes. Now, the reason we look at donovani is because it causes visceral disease, and at the time, GSK, that was their focus. Ultimately, we're really looking at causes of cutaneous disease. So azenic amastigotes, what are these? These are amastigote pathogenic forms which are cultured outside the host cell, so they're easier for us to deal with. So we screen the 211 compounds against these azenic amastigotes, and our hit criteria are EC50, an effective concentration that kills 50%. It's a log value greater than 5. So essentially 10 micromolar or less EC50. Property forecast index. That is a combination of physical, chemical characteristics of the compounds, and we look for a number that's greater or equal to 8. Aromatic rings related to the PFI, less than or equal to 4. and of course selectivity. We're using HepG2 cells. Again, it's a log factor. So we're looking for selectivity above 10-fold in relation to this classic liver cell line, which is used for toxicity studies. At that point, we've gone from 211 to 25. And then these 25 go into the rather more labor-intensive, complex intramacrophage screening. So we see here the macrophages of Leishmania donovani, sorry, the amastigotes of Leishmania donovani within macrophages. So using a fluorescent camera and some smart software, which was out of our hands. Again, similar criteria, apart from the fact the selectivity is against these macrophage THP1 cells. So of the 25, we have 5 hits that reach these criteria. So quite quickly gone from 2 million to 5. There's 2 series, 2 pairs of related compounds and 1 singleton. And for various reasons, we focused on these benzazepanes we can see here. Now, very important, we've gone through a cell-based system in the yeast. We want to prove that it can hit the enzyme in vitro. So we do have a complex, very laborious in vitro a platform for looking at IPC synthase activity. And indeed, the identified benzazepanes and some of the derivatives we got access to all had IC50s less than 1 micromolar or about 1 micromolar. So the assay worked, and we published that. However, none of them actually showed inhibition in an animal model, not significantly anyway, not at the level on oral therapy that we needed to demonstrate. However, working with our colleagues in Latin America, particularly Sylvia here, you look at tamoxifen is also an IPC synthase inhibitor, and indeed has actually even gone into some clinical trials for leishmaniasis in Brazil. And more recently, working with Patrick Steele and Bartira Rossi-Bergman, both key members of our NTD network, we've looked at clemastine fumarate, which is an antihistamine. And that has shown, at least under some forms of administration, the ability to prevent the formation of lesions in the mouse model at a level rather equivalent to an antimonial. Now, this is all open access, so you're very welcome to read these papers. But I want to use this to really emphasize the use of a technique called metabolomics, where we use mass spectrometry. Again, this is in collaboration with Mike Barrett. And we look at the levels of various factors that are associated with IPC inhibition. Now, clemastine was initially identified as an IPC synthase inhibitor, the inhibitor of the Leishmania enzyme. And if we look at the levels of metabolites, we see that ceramide goes up, for example. Now, ceramide is a substrate for IPC synthase, so inhibition would theoretically lead to an increase in ceramide, which it does. And also within this, and I'm not showing the data, we also managed to create resistant mutants, only 3 or 4-fold resistance, but these also had genomic effects, which we could trace back to an on-target activity. So this is the kind of proof we need to demonstrate that a compound is on target within a pathogen. Okay, that's all I'm going to say about yeast-based screening. Now, in general, the screening in industry, at least for this period, has really focused on target-based in vitro screening. Phenotypic has been a significant portion, but a smaller one. But then when we look at the ones that identify first-in-class compounds, new, genuinely chemically novel compounds, we can see that well over 50%, this is kind of reverse of this pie chart, are identified by phenotypic approaches. And this is where our colleagues at GSK really came into their own because of their abilities and their technology in actually working with academics to screen phenotypically their library against the 3 kinetoplastid parasites in various systems, Trypanosoma cruzi, Trypanosoma brucei, and Leishmania donovani. So this produced a number of hits. Now, also in 2015, this was a rare and exciting time for us in parasitology. We had Campbell and Mura and 2 winning the Nobel Prize for physiology or medicine for the discovery of Campbell and Amura of avermectin, which is been proposed to be an anti-COVID drug, but is more commonly known as an anti-parasitic, and UU2 artemisinin, natural products, now frontline drugs. Leishmania, current treatments also use natural product anti-parasitics, amphotericin B and paromycin, which I mentioned at the beginning. So using a phenotypic approach, again working with a company. This company is Hypha Discovery, which could be not more different from GSK if you try to design a company that is more different. It is small. It has a small, tightly held intellectual property portfolio and a low bureaucracy. So it was a different experience. It had its challenges, but through very little funding, actually, we got access to a small library of fungal extracts, over 300. And these extracts are secondary metabolites and they're low in molecular weight, less than 500. So they are in the ideal space for medicinal chemistry development if we can find anything that works. And we chose a platform where we'd look at Leishmania mexicana azenic amastigotes. Why use Leishmania mexicana? Well, it's a cause of cutaneous disease in Latin America, and also it's relatively straightforward to manipulate. So we get these host cell-free azenic amastigotes, and we look at some mammalian cells, monkey cells, actually vero cells, for cytotoxicity. So screening these, we get 38 potent hits, okay, against the amastigotes, and 23 have a very high selectivity index. So we're down to 23 at that point. And final selection after we talk to the company and they subtract those for known cytotoxicity, IP issues, and dereplication. The compound mixtures that actually have been shown to be active before, we get down to 12. Okay, now it's important to emphasize at this point that these 12, these are extracts, they are complex mixtures of secondary metabolites. So we don't actually really know, as the academic in this relationship, what's in them. So then we go through primary fractionation by high pressure liquid chromatography. So just after the first fractionation, and we're fractionating each of these 12 into 24 different fractions. And this is growth. So the clear patches are where we have hits in the assay. So we have the identification at this point, even on the primary fractionation, of striatal B, which is previously reported to be an anti-leishmanial. So this is our proof of concept. We are identifying anti-leishmanials. And then we go through the secondary fractionation. As you can see here, again, we go through the same thing. We are fractionating these into multiple fractions, 24, 25 fractions, and we have very few that are active. Okay, and all of those that were active and were taken forward to a third round of HPLC fractionation, all activity was lost. But at that point, we have one hit. So really, it's a very aggressive process. We've gone from 313 mixtures to one pure compound. And it's this fraction 11, fraction 9 of HGLSX27. So obviously, activity against amastigotes is good, but we want to look also at activity against infected macrophages and uninfected macrophages, because we want selectivity. So the activity against infected macrophages was great. Actually, it was approximately the same in terms of the EC50 as we saw in the azenic form. So that's excellent. However, the uninfected EC50, whilst it was considerably less toxic against the mammalian cell, it was a fairly narrow selectivity index of 5.5. So we would seek this, as I mentioned before, to be over 10. However, we did look at the structure through mass spec and NMR, and we have this novel compound, sesquiterpene, with a lot of oxygenation and unsaturation, which pretty much told us it wasn't really a drug that we could develop. However, we did look at the mode of action, and I want to sort of mention this because this is the kind of approaches we're taking. This is working with Mike in Glasgow, Mike Barrett. So essentially, we treat our parasites with twofold the EC50, and we look at the changes in metabolism we see. And the most significant changes, the ones that are up, cysteine, you can see here, is up significantly, and trypanothione. Okay. Now, so trypanothione is diminished by almost entirely in the cells, and also the precursor glutathione is also more than 60% down on treatment. On the reverse side, the oxidized cysteine derivative, cysteine, is only identified in treated cells. So all of this suggests oxidative stress. However, I should add, the peak resolution, the mass spec peaks, was quite poor. So we could not really be 100% certain that this simple mechanism was the absolute one. Okay, so Leishmania and other trypanosomatids, kinetoplastids, have a trypanothione peroxidase system, and central to that is trypanothione. Now, the molecule we identified has two alpha-beta unsaturated ketones, which you can see on either side of the molecule. And these have the potential to act as Michael acceptors. And this really gives them a highly reactive nature towards nucleophiles such as trypanothione and indeed glutathione. So our theory is, although we haven't got full proof, is that we're mopping up the trypanothione and we're upsetting this peroxidase system, and essentially we're removing the ability of the parasite to deal with oxidative stress. So we published that, and also we're looking at saponins as well, semi-synthetic saponins. Very, very different. These are likely to be membrane intercalators. Now, the important feature of this one is it's non-toxic towards mammalian cells. Okay, so there's a number of things we've covered there. screening platforms, both target-based, yeast-based, phenotypic, and also talking about deconvolution, which leads to my last slide and the project we're currently engaged with, a project that includes Iqbal amongst others, with Brian, Edu and Laura, the workers in Durham. And we're working again with GSK with funding from the Tres Cantos Lab Foundation and our Global Network for Neglected Tropical Diseases. And these are 15 compounds from the phenotypic GSK screen I mentioned a few slides ago. There's no indication about how they work. So we're using such things as the genomic approach I mentioned for clemastine, where we look for mutations and changes in the genome associated with resistance. We're going to use metabolomics. We're going to look at the lipid profile as well. And also we're going to use something called thermal proteomics, which will directly indicate the binding of the compounds to proteins within the parasite. So using this, we really working towards getting an idea of how these compounds work, both against the leishmanial parasites, but also against Trypanosoma cruzi, which cause Chagas disease in Latin America. Hence, we're working with Carlos and Santuza. Now, all of this, of course, is not done by me in the lab. It's done by these guys. So thanks to them, as well as my Durham collaborators, all of whom are within the network, and a whole list of collaborators, both academic and industrial, across the world. So thank you very much. I'm very happy to answer any questions. So I'll just stop my slide share if I can. Can I do that? Pause share, does that work? Stop share.
Thank you very much, Professor Paul, for very well put and a comprehensive lecture. With your permission, can I take some questions posted in the chat box for you?
Of course, I'd be very happy to.
So we have first question from Chris. He's asking, how have supply issues and high demand for liposomal amphotericin B affected treatment rates?
Yeah, that's an interesting one. Now, currently, it's supported by WHO and working with Gilead, which has not got the best reputation as a pharmaceutical company, to be honest, but it is working with WHO on this. I think there have been supply issues, but I think it has, at least at the current point, not interfered with the elimination program that is happening in India, Nepal, and Bangladesh. But I would stand to be corrected on that. I'm not intimately familiar.
So expanding on that, he's also asking, is it similar to other diseases such as Cryptococcus, where in affected areas it tends to be unavailable owing to high cost?
Yeah, well, I think, yeah, I mean, because of the support of WHO for the liposomal formulation, and it is a coordinated global plan, the costs are reduced a lot. Now, I suppose the concern would be into the future is whether that support can continue. Obviously, if the elimination program, this is solely for visceral disease, is successful, then that is not necessarily a problem. But I suspect that in the absence of other effective therapies, there'll be a need into the future for liposomal formulation of amphotericin B. And I am aware that the problem with Cryptococcus is cost and distribution.
Right. So we have another question from Ms. Sadra. She's asking, what can be the natural potential sources that contain active compounds which could be effective against leishmaniasis?
Well, I mean, that's a chicken and egg question, really. Now, what I would say is the most important thing, if we're going to develop any natural product, and it really could be from any source. I think that's, Samara, as you know from your own experience and work at ICCBS, is that looking at ethnographic information and known herbal therapies can be very effective. And go back to UU2 and artemisinin, that was basically from identified from Chinese medicine. However, the artemisinin is as good as it is because it's been identified and purified. It's not synthetic, it's semi-synthetic at best, but it is a defined compound. So whatever source we seek, we should look for the chemical entity that is responsible for the activity.
Right. We have another question from Mr. Muhammad Yunus. He's asking, apart from the drugs you have mentioned, can you give some info about methotrexate targeted against DHRFR? Is it in process for clinical trials?
I can't comment on that because I don't know, unfortunately. But of course, you know, that is a well investigated drug target with at least 4 such things as toxoplasma and plasmodium with drugs on the market, but I don't know the particular details of that.
All right. So here is another question. Is there any relation between anti-leishmanial drugs and production of ROS, reactive oxygen species, from the parasite itself or the host cell?
Yes, there is. The antimonials certainly have this effect. Again, by affecting that trypanothione-based redox system, and although they do have multiple other targets and effects in the cell, and even in our experience, the clemastine, for example, has and I didn't show this data, has a host effect that actually leads to a ROS antiparasitic effect as well. So it's not solely the targeting of a protein within the parasite that leads to the antiparasitic effect, it would seem.
Right. So here is another question from Ms. Misbah Rasool. She's asking, is nanoparticles like liposomes are proved more potent or efficacious than the conventional drugs for the leishmaniasis, as you told amphotericin B liposomes?
Yeah, I think obviously there's a lot of work on these nano formulations in academia and industry, and they could well, in many instances, I'm not a pharmacologist, but they could overcome some of the pharmacological issues we have with various antiparasitics and their administration. I suppose, again, it comes back to the question about liposomal amphotericin B and cost and supply. I mean, they can be and remain quite expensive formulations. And we have to be really aware that when we go through a discovery process where our target market, and I don't like to use the word market because this should all be based on charity, but our target market, our target patients are generally the poorest in the poorest countries. We must make it affordable. We also must make it very easy to transport and administer. So when I was speaking to the doctors at the Jinnah Hospital who were actually treating the people, I did ask them, what mode of administration would you prefer? And I was told the first would be a tablet, the second would be a cream for the cutaneous disease. Probably we need both, but it has to be affordable.
Right. So we have a set of questions from Mr. Nathaniel Jones. He's asking, how important have structure-guided drug discovery approaches been in your hit expansion, that is hit to lead optimization? Have your targets been traceable in a crystallographic assessment? In absence of this, do you use other techniques to gain structural insight like NMR, etc.?
Yeah, thanks, Nath. That's an interesting question. And I think Nath will correct me if I'm wrong. It's probably precipitated partly by the DeepMind production of predicted protein structures for Leishmania infantum and several other pathogens, where we have structures where we can look at structure-guided drug design. There's no example, which won't surprise you, for leishmaniasis for this, and the number of examples in the on the market for other conditions are small. I did ask a student this question as I was examining them. She didn't know either, but there's a small number. There's a few very prominent ones. But I think if the DeepMind structures, the predicted structures, turn out to be informative, which we really don't know at the moment, then maybe the use of this technology will be applied more and more readily for leishmaniasis. But of course, we do need well-validated targets, which is our raison d'etre.
Right. So we have another question from Dr. Samar. What is hope for vaccine against various forms of leishmania?
Well, obviously, everyone's talking about vaccines now for the first time in probably 60 years or longer. There are a small number of groups coordinating around the world looking at vaccines, including Paul Kay at York. And I think the hope is that logically a vaccine for leishmaniasis should be possible.
Thank you very much, Paul. There are actually a lot of questions in the chat box, but due to shortage of time, we cannot take each one of them. But I hope that your talk and your question answer session have covered most of them to some bit. And with this, I would request His Excellency, Professor Dr. Iqbal, to say a few words about the session.
Thank you very much, Professor Denny, for an outstanding lecture. It's not only the good science, but the way you present it is absolutely phenomenal. The way you have built the whole structure of presentation was more of a story-like, and we like stories, And so it was extremely coherent with an excellent flow of event and how the entire concept of developing drugs emerge in laboratory and how you perceive the whole thing. One more important thing which we truly are impressed with, that your realistic approach of following both phenotypic as well as target-based, because very often people just follow one of it and then they lose the opportunity of working on the other very important aspect of it. So in genuine sense of the word, it was splendid and an excellent presentation, which I'm sure everybody benefited. And let me tell you that it's not so easy to keep 200 people online all the time till the last minute, so close to 200. Thank you very much, Paul. Thank you.
Well, thank you, Iqbal, and thank you, COMSTECH, and thank you, everybody that tuned in. I enjoyed it, and I hope it was useful. So thank you very much, and good morning, good afternoon, good evening. Enjoy the rest of your day, however much is left.
Thank you very much. Thank you very much to our participants for joining, and for everyone who's asking for the recording of the lecture. This lecture is recorded and will be available on COMSTECH YouTube channel, which is by the name of OIC COMSTECH. So you can access it there as well. And thank you very much again to Professor Paul and to Dr. Samar, Dr. Henna, and Professor Iqbal, and the participants for being with us today. Thank you very much, and bye-bye.